just going to have to very quickly switch over. There we go. So I start off just to introduce myself. My name's Alice. I am known in the skeptical community through my work with the Merseyside Skeptic Society, so I'm a board director of the Merseyside Skeptics. I've been part of the Skeptics with a K podcast for about 3 years now, and I also have a blog where I write about topics like cancer and various other different sciencey things. But my other passion has been in kind of hardcore research science, which I've been doing for quite a long time. So I've been doing a PhD in cancer research, and then I did a couple of postdocs in cancer research as well. And then very recently, I've just switched over to a department of pharmacology where I'm working on both HIV research and nanomedicine research. So a slight sidestep. But I'm very interested in how cancer works and how cancer cells and how the cell biology of cancer cells works. And I think it's an area that's quite important. So I've decided that I would write a talk. So whenever I say to people that I'm a cancer researcher, one of the first questions that you always get asked is, okay, have we cured cancer yet? Are we ever going to cure cancer? People want to know about the cure. It's not a very easy question to answer as a cancer researcher, so I very frequently have to shorten the answer and say, well, actually, it's quite complicated. It's very hard to know whether we're ever going to cure this disease. So after trying to shorten the answer down a lot, I decided I would write a full-length talk. So to start off with, I want to talk about what cancer is and how it works, and then I'll talk a little bit about how we treat it. And then that feeds in quite nicely to the cancer pseudoscience, which has that kind of skeptical angle, but it's also such a massive topic within the field of cancer research that a lot of people are looking at alternative therapies for cancer. But just to touch on why this is important to us, you might have heard, and this is data from a charity in the UK, Cancer Research UK, where they have shown that 1 in 2 people born after 1960 will be diagnosed with cancer at some point in their lifetime. So this affects a huge proportion of the population. Not only does it affect a huge proportion of the population, this has increased quite drastically over the last few decades. So people born after 1930 had about a third of a risk of being diagnosed with cancer over their lifetime, and this has shot up to over 50% of people now. And there's actually quite a big difference between men and women. So men are more likely to be diagnosed with cancer in their lifetime, and women are slightly less likely to be diagnosed with cancer in their lifetime. So to talk about what cancer is, the most important thing first is to understand what the human body looks like. What does normal look like? So hopefully it's no surprise to anybody that the human body is made up of organs. And organs are made up of cells. So we've got lung cells within the lung, liver cells within the liver, different types of skin cell within the skin, and skeletal muscle cells in the skeletal muscle. And in fact, we have 13 trillion cells in the human body. It's a huge number. So this is just a list of cells that are specialized to release stuff, to secrete stuff other than hormones. So not only do we have 13 trillion cells, we have so many different types of cells that only the ones that secrete things other than hormones is a list this big. I couldn't put the hormone-secreting cells on a slide because it was far too long a list. So this is quite a specialized group of cells. These are cells like the salivary gland cells, cells of the mammary glands, cells that secrete mucus or things that aren't hormones, which is quite a niche specialism, and yet there's still this many different types of cells. But broadly speaking, every cell within the human body looks something like this. There are a couple of exceptions, but largely speaking, they all have a membrane around the outside of the cell, a nucleus in the middle where the DNA sits, and then they have a group of mini organs or organelles. So these are the things that allow the cell to do its job. So you've got things like the mitochondria, which generate energy for the cell, and things like the lysosome, which is kind of like the cell's stomach. It's an acidic compartment that breaks things down and allows the cell to reuse them. So humans are made up of organs, organs are made up of cells, and because we have so many different types of cells, they all have to do a lot of communication between them in order to allow the human body to both develop and to exist. So they do lots of different things. They have to know what type of cell that they are. You want your liver cells to know how to be liver cells. They need to understand what a liver cell looks like. These cells need to know how big to grow. They also need to know how many of them to grow. So this is proliferation, when you have the division of cells and the growth of cells. They need to be able to communicate with one another. This is particularly important in things like the heart, where all of your heart cells need to know to contract at exactly the same time. If they don't contract all together, you don't get this pumping action. They need to know where they are. It's all very well and good, your lung cell knowing that it's a lung cell, but if it's not in the lung, then it's not doing its job very well. And importantly, cells also need to know when to die. So this is quite important in development. So this is an example of somebody whose cells haven't died quite properly. When you're developing in the womb, your fingers and toes have webbing between them, and those cells in between your fingers and toes die back, and they have to know how to do that. And this is also important just in general, cell human kind of development and sustaining human growth. So for cells to be able to communicate, they need to be able to have a way of communicating. And the way that cells communicate is through proteins. But in order to explain what proteins are, I need to explain a bit more about DNA. So I said before that we've got the nucleus, and this is where the DNA sits. So within the human genome, we have a load of DNA that's sometimes organized into 23 chromosomes, and along each piece of DNA, we have these organized regions that we call genes. And genes are basically recipes. So genes are made up of 4 letters or bases, A, T, G, and C. And these are actually organized into groups of 3. So 3 letters of your DNA makes up what we call one codon. During production of proteins, the DNA is converted from DNA to a very similar molecule called RNA, and it retains this organization of triplicates, of codons. And then each of these codons can be turned into an amino acid. This maps one amino acid. And the amino acids build up into proteins. These amino acids are the building blocks of your proteins. So you can have a very complicated set of proteins. And in fact, in the human genome, we have 20,000 genes, which means we have more or less 20,000 proteins. So that's 13 trillion cells, but also 20,000 proteins. The inside of the cell is a very busy place. This isn't 20,000 proteins. This is quite a lot of proteins. And you don't need to understand any of this slide. It's just to give an example of how complicated the inside of the cell is. So this is the cell membrane up at the top here, and this is the nucleus down at the bottom. And in between, we've got loads of proteins communicating with one another, passing on signals, passing on messages between each other. So here at the surface of the cell, we've got proteins that we call transmembrane receptors. They come across the membrane of the cell, and they can allow a message to come from the outside through to the inside. And then all of these proteins interact in a stepwise manner that gives a signal which can then be moved into the nucleus and allow the DNA that is turned into proteins to change. So we will get different sets of proteins in different cells responding to different signals. So it's a very complicated, very busy place. And this is all going on inside every single one of our 13 trillion cells. So now we understand a little bit about how complicated the human cell can be. we can start to think about what cancer is. And at its most basic level, cancer is quite simple. It's just an overgrowth of cells. It's cells growing out of control. But because we have so many different types of cell, we actually have many, many different types of cancer. So actually over 100 different types of cancer. And here are just some of the categories that we have. And each of these categories are broken down into smaller categories. So you've got things like leukemia, cancer of the blood, melanoma, skin cancer, cancer of the brain and spinal cord, and then we can start to break this down at smaller levels. But again, broadly speaking, cancer cells have a few things in common. So this is a schematic taken from a very famous paper from Hanahan and Weinberg, which was very recently republished. And they categorize cancer cells as having all of these things in common. These are the things that make a cancer cell a cancer cell. So they're able to evade growth suppression. Those signals in the body that tell cells to stop growing, cancer cells are able to ignore that. They can avoid the immune system. They don't get destroyed by the body's natural defenses. They're effectively immortal. They can replicate forever, which our normal cells aren't able to do. They promote inflammation. They're invasive. They metastasize. They spread around the body. They induce what we call angiogenesis. So this is the creation of new blood vessels. This brings a nutrient supply into the tumor. It allows the tumor to grow bigger because it's bringing in fresh blood into the system. They have genetic instability. They're very prone to mutations, and their DNA is constantly changing. They resist cell death, so they don't die in the way that normal cells would. They have slightly different metabolism to normal cells, so they get their energy in a slightly different way to normal cells. And they have this sustained proliferative signaling. So not only can they replicate forever, but they're constantly doing it. They're constantly replicating. And we know that cancer is caused by protein mutations. So mutations in the DNA that gives you a change in your protein. And this can be a mutation in one protein, in things that we would call either an oncogene or a tumour suppressor. So just to give an example of this, a slightly simpler cell signaling pathway. It's actually taken from my PhD thesis. So we've got a single receptor spanning the membrane, and then we've got two different pathways that can be activated. One allows the cells to replicate, and one allows the cells to survive. And within these two pathways, we have some proteins that are particularly important for activating the two pathways. So, for example, the receptor itself, and then these two proteins here are very important in activating this pathway that allows the cells to replicate and this pathway that allows the cells to survive. So these we would call oncogenes. They activate the pathway, and if they're mutated to make them more active, they promote cancer. They promote the replication that we see in cancer cells, and they promote the cell survival that we see in cancer cells. We also have proteins in here that actually dampen down the signal. They turn the pathway off, or they reduce the signal. So here we've got one called P10, which serves to reverse this pathway. It turns off the cell survival. And this can be mutated in cancer to make it less active. So if this protein is less active, then this pathway is more active. You've got more cell survival. So under normal conditions, this is a tumour suppressor, and when it's not active, it can promote cancer. But actually what we do know is true is that not only do we have one or two mutations in cancer cells, we can actually have many, many, many mutations. And this can be multiple mutations in one cell, or it can be across different cells within the tumour. So this is just an example of this. This was from a paper where they were looking at the number of different mutations in different types of cancer. So here we've got things like colorectal cancer, where there are about 750 mutations per tumour. So a huge number of mutations. Things like lung cancer here are down to about 150, just over, mutations per tumour. And then here in the middle, the adult solid tumours, we've got things like head and neck cancer, esophageal cancer, gastric cancer. we're down between about 80 and maybe 50 mutations per tumour. So still really huge numbers of proteins that are mutated. Down here we've got the paediatric cancers, which often have a lot fewer mutations per tumour, which is why we're actually pretty good at treating paediatric cancer. We know exactly what causes the problem, and we can focus on that specific protein. But for most tumours, there's a lot of mutations. And as I said before, this can be in a single cell. You can have different mutations, or you can have different mutations spanning different areas within the tumour. So if we know we've got all of these mutations, we might want to understand which ones are the most important ones. So this is a very recent paper that came out right at the end of last year, where they were trying to look at, okay, we know that tumours have lots and lots and lots of different mutations, but how many actually drive the cancer, make the cancer be a cancer? And they found that, on average, it's between 1 and 10 mutations per tumour. So still quite a large number, but much smaller than the full number of mutations. So we've got to try and pinpoint which of those 10 from that potentially 750, which ones are actually driving the cancer, and then we can use that to try and understand how to treat it. So, hopefully, that gives an idea of how complicated cancer is and how difficult it can then be to try and treat it. So the next question is, how do we treat it? So this is a list from the National Cancer Institute that has basically all the different types of treatment that we have categorized, and we can split this into two main groups. So we've got the general anti-cancer treatments. These are the treatments that are things like surgery. You're cutting the tumour out. It's not specific to that type of tumour, you're just removing it. Or radiation, where you're targeting all the cells in a specific area and trying to do something damaging to them. Or we can have drugs that can do this, that will kill any cancer cell. They're not specific to the type of cancer that you have. But as we get better at understanding how cancer works, we can start to have more specific anti-cancer treatments. So we can have specific drugs or specific activation of the immune system to try and target the cancer specifically. So if we take the example of oncogenes, we know that in many, many different types of cancer, these particular proteins are hyperactive, they're activated. And so we can use an inhibitor or a chemical compound to block the activity of these proteins. This is actually not something we've successfully done for this protein. This is nicknamed the undruggable kinase because we've never been able to find a drug that will inhibit this protein. But we have very good inhibitors for some of the others, and in fact, this is very commonly inhibited in breast cancer. Herceptin is a treatment that a lot of people are given for breast cancer. When it comes to cancers that have a mutation in the tumour suppressors, these are the cancers where this protein is less active. It's hard to give a compound that will make it more active, but we can understand the pathway and say, okay, this is reversing the activity of this protein here, so let's drug that one. Let's inhibit that one and do the job that this protein should be doing. And that works very, very well. In fact, it's working so well that we've got survival rates, 10-year survival rates of cancer up to 50%. Now, this has doubled in the last 40 years. It's quite a big jump, and this is despite the fact that there's a huge variation in survival across different cancer types. So some cancer types only have a 1% survival rate over 10 years, and some have a 98% survival rate over 10 years, because, we understand some cancers slightly better than others. But broadly speaking, we've managed to double the survival rate in the last 40 years. But cancer can be quite clever. And this is why I like to give a bit of a case study. A few decades ago, we were trying to understand how chronic myeloid leukemia works. So chronic myeloid leukemia is a type of blood cancer, and we know that in 80% of all patients with chronic myeloid leukemia, they have a particular mutation that creates this fusion protein called BCR-ABL. Doesn't matter what it does, doesn't matter anything about it, just it exists in 80% of patients with CML. Now, this doesn't exist in a normal person. These two proteins are actually on two entirely separate chromosomes. So you get a mutation that brings the two chromosomes together, and it creates a protein that wouldn't normally exist. And what it does to force the progression of cancer is interact with a substrate. Again, it doesn't matter what the substrate is, it doesn't matter what it specifically does, but this interaction is the important bit. So we designed some drugs. Very smart chemists used rational design to design a drug that specifically binds to this part, to the bit that recognizes the substrate. And it sits here in the active site, and it sits there irreversibly. It binds and blocks and stops this protein from working because it can no longer bind to its substrate. But I told you earlier that cancer cells are very prone to genetic mutation, and cancer cells very quickly develop a mutation, often the same mutation or within a couple of different mutations, in this area that stops the drug binding but allows the substrate to bind again. And so patients become resistant to the therapy. Because we understand how this works, we can then design second and third and fourth line treatments that patients can be moved on to as they develop resistance to the drugs. But we know that they're going to develop resistance eventually. So we have to be clever when it comes to treating cancer, and we have to kind of always be one step ahead. So I have two examples of how we're trying to be smart with treating cancer. One is reacting to the Zika virus outbreak that was kind of everywhere a few years ago. Researchers noticed that Zika virus, the reason it specifically affects babies developing is because it affects brain cells in a very particular stage of their development, which doesn't happen in normal adult humans. And so the Zika virus doesn't affect the brains of normal adult humans. But cancer cells, brain cancer cells, have activated these development processes that the Zika virus targets. So smart researchers have said, well, okay, can we make the Zika virus safe in patients, but because it targets specifically brain tumors or developing brain cells, can we use that to treat cancer? And there's a lot of research going on into trying to use Zika virus to treat very difficult to treat forms of glioblastoma and brain cancer. This is possibly my favorite example because this is not just relating to treating the cancer, but actually relating to minimizing side effects in patients. So a lot of chemical treatments that we use in patients to treat cancer will also make you feel quite sick. They are compounds that affect normal proteins in your cell. They're just proteins that the cancer cell has more of or more active versions of. So it's still going to affect your normal cells a little bit. So researchers have been looking into how we can minimize this by packaging up the drug inside little bubbles, nanobubbles, made up of little nanoparticles, so that it can pass through the bloodstream of the patient unimpeded. It won't interact with any of your normal healthy cells. When it gets to the tumour, the researchers can disrupt these nanobubbles using an ultrasound, which is very localized, very specific to the tumour site, and it releases the drug and allows it to pass through into the tumour and only affect the tumour cells. So it's really cool research, which has been done quite successfully in mice and is being moved on into human trials now. So that's a bit about some of the cool research that we're doing at the minute and how we're trying to stay one step ahead of this very, very complicated disease. It wouldn't be a skeptics in the pub talk if I didn't talk about the pseudoscience. But I'll start off with the science. So just using a bit of a case study, we can talk about cancer and the diet. So the diet is implicated in cancer a lot. You will kind of hear it all over the media that the diet is important for regulating whether you will get cancer, and a lot of people will claim that it can treat cancer. So this is an infographic, again, from Cancer Research UK, where they've put together a few tips on how to reduce your risk of cancer. And they say one of the most important things is to maintain a healthy weight, We know that obesity is very highly linked to an increased risk of cancer. But then another thing they point out is that you can cut down on things like processed or red meat. And this was actually related to a classification update from the World Health Organization a few years ago. You might have seen it in the media. So here's a few headlines from the UK media that we saw, where newspapers were telling us that bacon, ham, and sausages have as big a cancer threat as smoking. Processed meats do cause cancer. 2 rashers of bacon a day raises your risk of cancer at the same level as cigarettes. A lot of kind of big, scary stories. And this kind of highlights quite nicely a problem with the media, and how that can affect people's perception of what works in the body. Again, Cancer Research UK put together a nice infographic to explain the differences between lung cancers caused by smoking tobacco and bowel cancers caused by eating red or processed meat. Because the science is there. We know that eating red meat does increase your risk of cancer. But the question is all about relative risk. So we know that 86% of all lung cancers are caused by smoking tobacco. And lung cancer makes up almost 20% of all cancers. So it's a huge proportion of a very common cancer, such that if no one ever smoked, we'd have 64,500 fewer cases of cancer each year in the UK. Whereas, when it comes to bowel cancers caused by eating red and processed meat, we know that 21% of bowel cancers, still quite a huge proportion of bowel cancers, is caused by eating red and processed meat. But that only makes up 3%. Bowel cancers only make up 3% of all cancers. It's actually still quite a common cancer. Bowel cancer is one of the big 4. It's one of the big 4 types of cancer. But if no one ever ate any processed or red meat, we'd have 8.8 thousand fewer cases of bowel cancer per year in the UK. When we actually look at the lifetime risk, you can see the difference between these. So what we found over the lifetime is that if you don't eat red and processed meat, your risk of bowel cancer is 5%. If you do, it goes up to 6%. So you're getting a 1% risk difference, really. But it is important. We know that diet is important. But these kind of miscommunications between the media and the general public allows pseudoscience trying to claim that cancer can be cured by diet to sort of proliferate and replicate through people's perception. So I'd like to give an example here of Gerson therapy. I don't know if anybody has heard of Gerson therapy. Podcast listeners will have, because we've talked about it on the podcast a few times. Gerson therapy is starting to get a little bit more popular. I'm starting to see it in the media a little bit more. I've taken this summary of Gerson therapy from the Good Thinking Society's website. So they have quite a few articles on different types of alternative therapies that kind of give you a bit of a background to them, which is a really useful resource. And they point out that Gerson therapy is the hourly consumption of organic or raw juices, extreme use of supplements, and a daily schedule of up to 5 coffee enemas, which is not a fun treatment. Not only is it not a fun treatment, it's actually very expensive. So again, these are figures taken from the Good Thinking Society's website. They say to set up the Gerson therapy, it will cost £7,000 for a 2-week stay at a Gerson therapy clinic. Now, actually, a lot of them recommend that you should stay 2 to 3 weeks, so it can be more money again. £1,600 for a Gerson juicer, £400 for a hydraulic press, £650 worth of organic produce, £650 worth of supplements, £325 for a distiller, and £20 worth of herbal teas. And then there's an additional monthly cost on top of that of £700 per month on organic food, £250 per month of organic produce, £200 per month on supplements, £20 per month on organic castor oil, £7 per month on enema pads, and £100 per month of Gerson doctor consultations. Now, they recommend that you take this therapy for 2.5 to 3 years at this cost per month, ideally for the rest of your life, which may or may not be 2.5 to 3 years. So not only is this, quite a disruptive treatment to take, it's very, very expensive, but it is also quite dangerous. So the risks that, again, the Good Thinking Society points out are that coffee enemas have been known to cause infections, dehydration, fits, constipation, mineral imbalances, heart and lung problems, and those heart and lung problems can actually lead to death. The diet itself can cause dizziness, abdominal cramps, loss of appetite, diarrhea, and sickness. It's not a fun diet to be on. People lose an awful lot of weight on this diet. But for me, this is the big thing. This is the biggest risk of following things like Gerson therapy. Proponents of Gerson diets often claim to be able to treat and even cure cancer, and they discourage patients from seeking real medical advice. So these are patients who are being told not to listen to their doctors and to listen to the Gerson therapy proponent instead. So this first came to my attention when we at the Merseyside Skeptic Society saw in our local newspaper this story. So the headline was, "Man with cancer beats 8-month prognosis despite shunning hospital treatment." So this was promoted in the local media, that this young man who had been diagnosed with a type of lymphoma had decided not to follow the advice of his oncologist and travelled to Mexico for Gerson therapy instead. And this was heralded as a big success in the local media. So we at the Merseyside Skeptic Society wrote a letter to the Liverpool Echo saying that the miraculous claims for alternative cancer cures make for impressive headlines, which are doubtlessly seductive, but as a responsible publication, you have a duty to your readers to put truth ahead of sensationalism. By promoting these so-called cures without scrutiny, the Echo, the local newspaper, lends these dangerous quackeries the legitimacy of the publication's well-earned reputation and promotes clear misinformation to some of its most vulnerable of readers. So we got this signed by a load of cancer researchers at the University of Liverpool, and we did get this published in the print edition of the paper. Whether it actually made a difference to the stories that they write, I'm not convinced, because the following week we saw quite a similar story promoting a different alternative therapy, but we tried. And then a few months later, we saw the guy who runs a Gerson Center clinic in Mexico was coming to our local city, to Liverpool, to give a talk. So his talk was called Censored for Curing Cancer, and in his promotional information, he said that the Gerson treatment continues to be the most powerful and effective alternative therapy available for advanced or non-advanced degenerative diseases, including terminal cancer. So we went along to the talk. With hindsight, we wish we'd got it cancelled, but we went along to the talk. There were at least 70 people in attendance. Their head count put it at about 100. This is a photo from that event. That is me there, along with Michael Marshall from the Merseyside Skeptic Society and a friend of ours, Sarah, who's recently actually joined the board of directors at the Merseyside Skeptic Society. The ticket price is £20 to £30. And we established during the Q&A that many of the people in the audience were patients or family members of patients who were undergoing treatment for cancer. Either they wanted to go on to Gerson therapy, they were considering trying Gerson therapy, or they were already on Gerson therapy. The guy who runs this Northern Baja clinic in Mexico is Patrick Vickers. He is, by training, a chiropractor. So, cancer expert, obviously. And he made some quite astonishing claims during his talk. These are a few of the things that he said. So he said that we are treating advanced terminal diseases, not just cancer, but virtually every single disease we're successfully treating. And in fact, throughout both the talk and the advertisements, he promoted the Gerson therapy as a cure for cancer several times, which in the UK we have a protection called the 1939 Cancer Act, which we think that this directly breaches. So this act says that no person shall take any part in the publication of any advertisement containing an offer to treat any person for cancer, and this includes notices, circulars, labels, wrappers, other documents, or an announcement orally or by any means of producing or transmitting sounds in a public setting. So we think some of these claims are actually breaching the Cancer Act. Kind of tricky to follow that through because he's he lives in Mexico. He doesn't live in the UK. He said, I'm telling you folks that the science is so crystal clear and it's so perfect that it's indisputable, which I definitely dispute. But he also encouraged a lot of distrust in people who might dispute his claims. So he encouraged a lot of distrust in medical professionals, in friends and family of patients, in charitable or governmental sources of information, the NHS website, for example, Cancer Research UK. He said of his fiancée, because she stuck with the Gerson therapy, that's the only reason why she's alive today. She had absolutely no chance of survival using all conventional medicine. And this is actually a really important point throughout his talk. He warned patients that if the Gerson therapy doesn't work, it's because they've made a mistake. They've done something wrong in administering it. They are responsible. They've not followed this very complicated treatment perfectly. During the Q&A, he was asked what his take on conventional treatment was. His response was, it's incompatible with Gerson therapy. And then when he was pushed on it, he said that chemo is made from mustard gas, biological warfare. It takes you as close to death as possible, and after, the cancer comes back 10 times more aggressive. In fact, during the Q&A, a lot of patients asked about their specific cases, and he directly told patients to stop taking their chemotherapy, to not undertake clinical trials that they'd been recommended by the NHS. And not only that, patients are told that they shouldn't have any scans within the first 6 to 12 months of starting Gerson therapy. And within that time, the cancer can progress quite significantly, and patients aren't having the scans that will tell them what's happening. So it's incredibly, incredibly dangerous claims that he's making. He encouraged a distrust of medical professionals, other reliable sources of information, and even friends and family members of patients, and pushed a reliance on information from him, from the Gerson therapy clinics, from the scans and blood tests that they recommend, that when they do permit them. And they told patients, that they are responsible for any failure in the treatment. And on top of that, this is costing thousands of pounds for patients. But that's just one type of complementary and alternative medicine. There's actually an awful lot of them. This is a list taken from Cancer Research UK of just a selection of them. I actually fairly recently put together a list for the Good Thinking Society, where we looked at different types of alternative medicine that people were fundraising for on big fundraising websites, things like Just Giving. So these are ones that people are actively fundraising for, and we put them together in a big list and explained a bit about them. There's all sorts of weird and wonderful things on here, including my favourite, which is dowsing. You can douse your cancer away, apparently. All sorts of things that if people are interested in specific, if you spot one you're particularly interested in, we can talk about it in the Q&A. There are things on here, quite interestingly, that are real treatments. Things like the PD-L1 vaccine is a very interesting new type of treatment that only works in a very specific set of patients. And so alternative therapists will claim that they can use it to treat patients, but then not test their patients to prove that they are the specific set that it will work for. And so it can be entirely useless in the patients that they're recommending it for. But then there's all sorts of things like apricot kernels and things that we know definitely don't work. MMS, which is bleach. Patients are told to take bleach to cure their cancer. And this one, scalar technology, which doesn't exist. It's a made-up type of energy which is used to treat cancer. Very bizarre. So hopefully, throughout this talk, I've told you that cancer is incredibly complicated, and we don't fully understand it yet, but we need to use the understanding that we are developing to try and treat it using evidence-based medicine that we can prove is reliably efficacious, and we can consider drug resistance and how the disease progresses. But what I've also told you is that cancer pseudoscience is pervasive, it's very dangerous, and it can be hard to detect because the media representation, even of the science, can be quite difficult to understand and to follow, let alone the pseudoscience. So that leaves me just to tell you where you can find me. These are my various contact details. I'm on Skeptics with a K, which is a skeptical podcast. I have got a blog, that is my old logo. I've got a new logo. And so these are two resources that I think are really useful. Cancer Research UK has these fantastic infographics. They explain cancer in lay terms, and they have some great, they compile loads and loads of statistics, so it's great to find out information about specific kinds of cancer. And then, as I said, the Good Thinking Society have some great summaries of how different alternative therapies and how they claimed to work and the evidence behind them. And that's me.